Well, welcome and thank you for joining us today. I am Dr. Carlos Bosquez, CEO at FOSSI, and it is an honor to welcome you to another great discussion within our seminar series. As the leading food allergy research organization, our focus is to understand, translate, and intervene with food allergies. And one key question driving our research is why has there been such an enormous increase in food allergies over the last few decades? So in September, we started a new seminar series that we call Project Safe, the impact of chemicals in our food and the environment. And the series highlights the science on how the evolution of chemicals in food and the environment could have an impact on human health, and especially how this may help explain the rapid increase in the development of food allergies over and other chronic disorders. So we hope you continue to join us for our monthly seminars where we spotlight the critical science around this topic and learn from experts in the field. And we have one more seminar left next month. And so it has been an exciting series. I hope you join us for that last seminar of this series. Today, I am thrilled and honored to be joined by one of our members of our scientific advisory board, Dr. Beth Sadley, from who's a professor, so the professor of chemical engineering at Stanford University, and she's also a Howard Hughes Medical Institute investigator. So Beth and her research is inspired by human reliance on plants, plant-derived molecules for food and medicine, and her research has been discovering and engineering plant metabolite pathways to make molecules that can enhance human health. And so it is an honor to have Beth with us. Before we go into the discussion, I just want to remind people that we're going to be taking questions from our Q&A box in the Zoom. So please feel free to submit your questions so that we can engage in after the discussion and answer your questions. Beth, it is an honor to have you here. I always enjoy your research so much, and I'm looking forward to your discussion.
Great. Thank you so much, Carlos. Thank you for the introduction. I'll get my slides shared here. Okay, can you see that all right?
Perfect.
Excellent. Okay,
Perfect.
let me get started. So I actually am going to start today with some personal photos that really remind me of the fascinating ability of plants to grow into some of the largest and longest living organisms on Earth. They do this in the face of all the same challenges that we have: the need to cope with pathogens and predators, the need to acquire nutrients and deal with environmental stress, but all without the ability to move as they're fixed in the ground as sessile organisms, without circulating cells, or without an adaptive immune system. So a fundamental way that plants achieve this feat is that they're multicellular organisms for sure, yet they haven't fully embraced centralized systems. And instead, they have many of their core functions distributed across the plant body. For example, a tree could be covered in hundreds of sites for seed distribution, or could exist as a clonal colony with new trees sprouted straight from the roots. Despite this apparent decentralization, plants still have the ability to respond and function in a very coordinated way. And from our perspective, how plants solve almost all of their problems is with chemistry, and that is the focus of my research program. As I hope to be able to convince you today, we think that plants are some of the best chemists on the planet. So just to give you an idea of some of the scope of all the different molecules, specialized molecules, in fact, that are made by plants, it's predicted that there's over 200,000 different unique molecules that can be found in the plant kingdom. But despite this incredible diversity, we actually know very little about what these molecules do and where and when they are made. So how does this relate to diet? When you take a look at this picture, you might be thinking about a food you either enjoy or something you despise, or maybe just an ingredient for your next meal. But when I look at a tomato, I'm thinking about it through a very different lens, and that is through, the chemical complexity that exists in this food, where many different types of molecules actually comprise a tomato. And we wonder, in fact, how do these molecules contribute to the nutrient profile and to the function of these foods as we consume them? What this means is that when we think of a typical nutrition label for a food, it's likely that these categories, total fat, total carbohydrates, and fiber, are overly simplified and don't really capture the chemical complexity of food and how these compounds influence the human health. Let's take fiber as an example. This is not one thing, but many different types of molecules with very unique structures that have different ways of interacting with targets in the human host. Even if you're not following the chemistry, you can track all the different colors of these rings to illustrate all the diversity that comprises fiber that's found in our food. Okay, so let's think about, we have all this chemical diversity of food. What does that mean for our diets? These are some photos that I took from a really fascinating book, Hungry Planet: What the World Eats. And in these pages, the photographer of the book has captured what a typical family in different regions of the world consumes for an entire week. On the left, you can see a family in the US, what they might consume, juxtaposed to a family in China having a very different composition of diet. And so the big question here is, how do these diets and the molecules they contain affect our health, and also the prevalence of food allergy in each of these different regions of the world and in all these individuals? So one thing that might not be top of mind when you look at what's on the table for each of these families is that these foods actually represent one of their largest exposures to the environment. So let's take a closer look at that. As humans, we encounter on the order of 0.5 kilogram of plants per person per day. The gut, of course, is the interface for which we interact with our food. But this is not a very small compartment. In fact, what does it look like? So when we think about the size and the scale of this interface for humans, for example, in the small intestine, the area is on the approximately the size of a singles tennis court. So all these folds and microvilli in your small intestine allow for this incredibly high surface area. And this is where there's an incredible amount of food absorption, nutrient sensing, and essentially perception of our environment. So what is doing the job? What are the ears of the intestine that can listen in to these food molecules from the environment? There's essentially two different kinds of things lining the tennis court. For the purposes of today, you can think about one as being channels, selective channels through which nutrients can travel. And the second thing is protein sensors that line this surface. There's many different kinds of sensors. They detect the small molecules and nutrients that are in foods, and then are able to transmit these signals inside into the body, resulting in the secretion and accumulation of different hormones that change how the host responds to this food. So this is just a table of some of these different types of protein sensors that line that tennis court. But what I really want to draw your attention to is the number of different types of proteins that can respond to food. So hundreds of different individual types and then many copies of each are what is essentially covering that tennis court, just waiting for the food that encounters. So that's essentially how we perceive food. But what is it coming in at the other end? What is the complexity of the food that we eat? So let's take a look back at those diets that I showed you for a typical family in one part of the world. On the left side, what I'm illustrating is many of the different chemicals that are accumulating in our food and that we encounter through our diet. For example, this molecule from soybean, which we might encounter on the order of 20 milligrams per serving per day, or these compounds from broccoli or apple or orange. So this might not mean very much to you, but I want to juxtapose the amount of these drug-like molecules with a typical molecule that you might encounter as part of a prescription. For example, Lipitor, which is prescribed on the order of 10 milligrams per day. So what this means is that these drug-like molecules in our food we're encountering at about the same levels that we might a pharmaceutical that we take every day. And so we're just beginning to scratch the surface in terms of how do these compounds that are present in our food influence the host and what is their role and function. So we know that they accumulate at high levels and we encounter them at high levels, but what are some of the possible roles? Again, we're just beginning to understand this, but I want to give you just a few examples of what we do know for how plant molecules can influence human biology. One leading example is this compound or this formula of compounds called QS21 that's found in the Querqualia tree. This molecule is incredibly complex, as I've illustrated with all these different colors, but it actually serves as a really potent adjuvant and is used in the formulation of different vaccines. So, for example, Shingrix, if you've encountered this vaccine, contains this molecule. And it has a really potent ability of being able to very specifically stimulate the immune system so that this vaccine can protect you from a shingles infection in the future. So this is just one example of some of the functions of plant molecules. Perhaps one that's a bit more traditional is the isolation of a molecule from a plant and then its use as a therapeutic. And one of the most potent ways that plant molecules have been used is as chemotherapeutics. So this compound, paclitaxel, or Taxol for short, is used in over a third of different breast cancers and many different cancer chemotherapy regimens. This compound comes from the Pacific U, and it has the potent ability to essentially modulate and modify the skeleton that's within cells, limiting cell growth. And so in this way, this plant compound can have a very potent effect in the context of a disease treatment. But it doesn't just have to be, again, molecules that are isolated from plants that can affect human biology. This is an example of a compound that many of us have encountered if we enjoy consuming brassicas, such as broccoli and cauliflower. It's a really interesting form in that this one serves as a prodrug. However, once you cleave off the sugar, it turns into an active form in some of the different examples I have illustrated here. This one on the left, for example, can be targets proteins that are important for pain perception. The one in the middle is known when in human trials to reduce fasting blood glucose. And it also is perceived by different proteins that are part of the cellular stress response. The one on the right has yet a different function where it can target receptors in immune cells. And so this just gives you an example of some of the pharmacopoeia that can come within the food we eat and how it might modulate human biology. The last example that I want to share is one that we're very familiar with, compounds from plants that modulate neurotransmission. And so I didn't choose caffeine for the example here. Instead, I'm illustrating a molecule from the club moss, which actually is a compound from plants, but it very much mimics human neurotransmitters. And so you can see by these yellow circles how it starts to look a lot like acetylcholine, which is a critical neurotransmitter that enables signals to transverse neurons at the neuromuscular junction. And so taken together in these four examples I've shown you, hopefully I've illustrated that there's many different roles that plant molecules can play. And it actually, from my perspective, starts to feel like quite a bit of complexity. So how are we ever going to be able to pick apart all these different activities and possible ways plant molecules might influence our health? And so what I want to argue today is that perhaps even though there's so many different molecules, we can start to identify a unifying logic for how these compounds work. And our proposal is that these compounds, the target space is not infinite, and instead there's themes for how these molecules might function. So let's take that example of neurotransmission as an example. Here we are back at the neuronal synapse with a neuron excreting a neurotransmitter that's perceived at the neuromuscular junction. I illustrated before a molecule that can interfere with this process from the club moss. But in fact, there's many different molecules that have evolved across the plant kingdom to target neurotransmission. Here's a second one from daffodil, and here's a whole bunch of different molecules, in fact, that can interact with many different ways that neurotransmitters are perceived in this process. There's two things that are really interesting here. One is that even if you're not following the chemistry closely, you can see that these are different molecules. They're very different shape and form. The other thing is that they come from all across the plant kingdom. So one of the compounds I just showed you is from daffodil. Another is from the calabar bean. This one down here is actually accumulates in potato, and we know that it accumulates in the context of human serum. So what this means is that neurotransmission has been a sort of a hotspot for evolution across the plant kingdom. And we can find many different variations on a theme that target this central player. And so to close, I want to just share some of what we start to think of as some of the themes of function that will help us start to simplify some of the complexity of all the molecules that we encounter in our diet. The first one, again, and this is just a version 1.0 of some of the themes, is molecules that have evolved in plants to target or modulate neurotransmission. We can think of a likely target here as animals, as because of course plants don't have brains. This is a completely orthogonal set of targets that could allow plants to manipulate animals that they come in contact with. A second set of targets that we find that's quite common are molecules that plants make that target really conserved processes in the cell cycle. I showed you Taxol before that is useful as a chemotherapeutic, and that compound targets the skeleton within cells. And so perhaps the evolved roles of those molecules is to target different microbes that might feed on plant tissue. A third example are compounds that actually might be used for communication, so might enable interkingdom signaling. So if a plant needs to signal, for example, to a bacterium that is living in the roots and helping it fix nitrogen in the context of these root nodules, it needs different molecules to enable that communication. Our fourth set of compounds fall under the category of molecules that just help plants do what they do, again, while being sessile organisms or rooted in the ground. For example, compounds that can leave the plant and help them acquire iron as they need in the context of the plant diet. A fourth category are molecules that modulate development. And for example, we think that some of these compounds here are important for targeting insects and limiting their feeding on the plant tissue. And the last category that I'll put up here as I'm thinking about what are some of these different groupings are just ways plants communicate with each other, just how they have conversations from one plant to another. So how do we think about these possible categories in the context of diet? I want to highlight that although some of the evolved targets for plant molecules might be insects, many of these pathways are conserved. And so in each of these categories, I've highlighted different molecules that can also target human proteins. And so there's some overlap here in terms of the possible protein targets for each of these molecules that we find in plants. And so taken together, I hope that this starts to simplify a bit some of the complexity that we see in plants and will help us begin to dissect the role of these molecules in our food. So taking you back to that tomato that I showed you in the first place. Again, what I want you to walk away from here is that this is not just a flavorful food, but really a bag of chemicals that has the potential to impact human health. And so I want to leave you with a few questions that we're asking going forward. How do these food molecules tune immune responses, especially to the proteins that are found in food? And this becomes particularly important when we think about why are we typically allergic to things like peanut and not to corn? What are some of the chemicals that might be in some of these foods that make it easier to form a food allergy to one food and not another? A third question is whether some foods are inherently tolerogenic and whether they could be used as a way to program tolerance and ultimately to prevent food allergy from forming. Are there certain chemical matrices that are interpreted as friendly by the immune system? And then lastly, as I illustrated to you on the slide before, what are some themes of plant molecule function? How can we start to categorize the different roles of foods that are found in our diet and think about them in terms of a logic of how they might influence human health and disease. And so with that, I'd like to close, and I'd be happy to take any questions.
Thank you, Beth. Thank you for an excellent presentation. Really enjoyed this discussion. And thanks for everyone for joining for this discussion. Before we go to the Q&A, people always ask us, how can they help? Here's a QR code. Donate, host an event, follow us on social media, join our network. And we are a nonprofit organization, so any support counts. So you can scan this QR code. Um, and just as a reminder, people should pay attention. We're going to have our last seminar on Tuesday, May 20th, and that's going to be Dr. Sophie Newton from Nestle will be joining us. So I hope people can join. So we'll move to the Q&A side. Beth, really excited, especially as you showed. Uh, one thing is we've been talking in these seminars a lot about negative impact of molecules in our food, but I'm so glad that you were able to provide examples and show how many positive impacts you can have from food and for molecules in the food that we consume, and the fact that even we consume them at similar levels that they're used for treatment. So that's fascinating. One thing that I was very intrigued, you put in the numbers of receptors that detect food in our guts, and I'm blown away of the complexity. We're putting so much food, tons in a lifetime, right, through our gut, and yet we have such exquisite sensitivity and selectivity. You show the fibers that have so many sugar molecules come together, and I'm blown by the fact that sometimes those sugars, if they're connected in a different form, like an alpha linkage of two sugars, that can lead to a reaction. Well, if it's not in a linkage that way, could be benign. So, um, can you tell us a little bit more about how do you see that sensing of those molecules coming through the gut, and how, for example, the microbiome may play an impact of letting us some of them pass through and some of them being, we react to them.
Sure. Yeah. Thanks for the question, Carlos. I'm going to split that into two parts. One that, and I'll start with your second question, actually, about the microbiome, which is an incredibly important component, because essentially that's a processing step. The microbiome can take these molecules and convert them into a very different form, and those different forms might have altered activity. So we have to think about very carefully. It's not just the food that's coming in, but it's what is the composition of someone's gut microbiome that might specifically alter the form of that food before it's presented to the human host. I think a lead example is that the set of compounds that I showed you from broccoli and cauliflower, those actually start out as being prodrugs, as I said. And just to explain what that means is they're not functional in their original form, but they have to be metabolized or go through a processing step. And we know that the gut microbiome can do that, and it can do it in different ways, leading to different downstream products. So it's very important, I guess, and my main point here is to think about how is an individual's gut microbiome processing and changing the molecules that we encounter in our diet. The other question you asked is this huge array of different receptors that are perceiving the chemicals that are in food, and how does that work together? We do think this is a very complex process in that there's many, many molecules that are being perceived at once. And so it starts to make you think about how can you get signal over noise with all of this complexity coming in. But what we understand is that these sensors working together are exquisitely tuned and very sensitive. And so they can listen into different molecules very specifically in different combinations and lead to different host effects.
Awesome. Thank you. Thank you for that. No, very, very interesting, all this detection. To follow up on that, there's a question here from Megan. Great talk. Love all the examples. How close are we to identifying the human receptors that detect plant compounds with immune response modulation, both adjuvants and tolerance encouraging chemicals?
I think right now, to be honest, I think we're at the tip of the iceberg in terms of really being able to match food molecules to receptors that are modulating immune responses. There are some examples for those broccoli molecules. In fact, one of those compounds modulates immune function, the one that I showed all the way on the right. And that one for sure is a dietary molecule. But there's other examples that we know from plants we don't typically consume that we know have the ability to modulate immune responses. I gave an example of the adjuvant from the Querculia tree. Another one that Dan Littman has studied is digoxin from foxglove, which can modulate the development of Th17 cells. Um, so there's evidence that these molecules have an effect, but we're still drilling in to what specific ones in foods have this role.
Excellent. Excellent. Um, I always wonder, in order to have such a selective process that our system won't react to so many molecules all the time, I wonder if there's a mechanism by which it's not one receptor detecting one thing, but you need 5, 10, 20 in different parts of your body to be sense or activated at the same time in order to detect. Is there examples where maybe there's example pointing to that you need so many different things at the same time converging in order to get a reaction?
I don't think we've been able to probe it in that level of detail yet. I mean, one thing that we do know is true that those receptors that essentially are lining the tennis court, so to speak, they're not static. And so if you're getting a lot of signal through certain pathways, more receptors can be placed on that surface, right? Or some of those receptors can be taken in so that you get less signal. So it's a dynamic listening process. And it changes, meaning it changes over time and it changes with the amount of molecule that is being encountered. So I think two things are for sure that there's the possibility of essentially combinatorial perception that multiple different molecules together can combine to have a single downstream effect. And there's also the idea that these receptors are very dynamic. So just to give an example, you can become acclimated to a certain food, right, or a certain molecule, just like, you might need more and more cups of caffeine over the course of your lifetime in order to get the same stimulant effect.
No, this is excellent. And Wolfgang, thanks for your question. I think we addressed the question of the microbiome and how the microbiome process food. Um, Thank you so much, Beth, for a great discussion. One last question is what's missing? What's missing for us, for scientists to be able to understand and then translate molecules from food to like examples that you gave, to therapies, to things that can be useful to everyone in the community.
Yeah, I think there's sort of two different things that we really need to do. One I sort of posed on the last slide, and that is in a very specific way, dissect two foods that have very different biological outcomes. So, for example, if you take peanut and you take corn, one is very prevalent in food allergy and the other one is not. So, what are the molecular differences between those two as sort of a case study to really understand how the molecules in those foods might be influencing how your immune system interprets the proteins they contain. So I think that's the first thing. The second thing is I think we really need to do very high throughput dissection of all the foods that we consume and start to categorize what are some of the possible roles that these molecules might have. Thinking about, those diets that I illustrated in the photo there, how do we move beyond just these are fats and calories and carbohydrates and really think about what are some more refined categories of molecules with specific roles?
Great. Beth, thank you so much. Thanks for great discussion. Thanks for everything you do for FOSSI as an as a board member. Really appreciate it for everything. And thanks for a great discussion. I hope everyone can join us in our last seminar next month. And thanks for everyone for being here today.
Thanks so much. Take care, Carlos.
Thank you. Bye-bye.